Okay, hello everybody. It's Dr. Kaur here. As we are not going to have the lecture on Thursday, so here is my video lecture. So this is your chapter number 5B. Let me go into the slideshow mode. Okay, so in the 5A we have already talked about the plasma membrane and how the transport takes place across the membrane. So now we are going to go inside the cell and we are going to take the tour of the cell, and inside the cell we are going to see different organelles. And you know what organelles are? Organelles are different compartments which perform different functions. So basically going back, we have already talked about what are prokaryotic cells and what are eukaryotic cells. So prokaryotic cells are the cells which don't have nucleus and they don't have the other membrane enclosed organelles. So eukaryotic cells, they do have nucleus and they do have some other organelles. So that's what we're going to talk in this chapter about the different organelles. So this is the prokaryotic cell, the simple cell, and this is the eukaryotic cell. We know the differences. We have already talked about the differences between prokaryotic cells and the eukaryotic cells. So your prokaryotic cells don't have the nucleus. Eukaryotic cells, they do have nucleus. And we have also talked about gene expression, and we know that in the prokaryotes, the transcription as well as translation takes place in cytoplasm, but in eukaryotes, transcription takes place in nucleus and translation takes place in the cytoplasm. And we know that prokaryotic cells are small in size and eukaryotic cells are bigger, and the prokaryotic cells don't have membrane enclosed organelles, but eukaryotes, they do have. So this is an example of an animal cell, and this is a picture of your plant cells. So both these cells are eukaryotic cells. So let's see what are the differences between an animal cell and a plant cell. So you can see here, this is your animal cell, and this, you can see inside the cell there are a lot of organelles. So this is the plasma membrane, and this is the nucleus, this is the endoplasmic reticulum, this is Golgi apparatus, this is mitochondria. And you can see that animal cells, they have cell membrane, they have a plasma membrane, but they lack the cell wall. So this is your plant cell. So plant cells, they do have the cell wall. This is the main difference between animal cells and the plant cells. Animal cells lack cell wall. Plant cells, they have cell wall. Plant cells, they have a central vacuole in which they store different fluids, water, some ions, etc., and some nutrients too. And also another difference between animal and plant cells is plant cells, they have chloroplast, and chloroplast is an organelle in the plant cells which has a special pigment called chlorophyll, and which is responsible for photosynthesis. So there are three main differences between animal and plant cells. Plant cells have cell wall, animal cells don't. Plant cells have central vacuole, animal cells don't have a big central vacuole, but they do have some small vacuoles. Then your plant cells, they have chloroplast, animal cells, they don't. So these are the main differences between the animal and the plant cells, and both these cells are eukaryotic cells. Okay, now let's talk about the cell. The cell is enclosed by a membrane called plasma membrane, and we have talked in chapter 5B in detail about plasma membrane, and you know its structure, and you know what is the function of plasma membrane. Things get transported in and out across the plasma membrane. So this is the basic structure which we just talked in the last lecture. It's made of a phospholipid bilayer, and these are the proteins embedded. Then we have cholesterol, which is not shown here basically, and the carbohydrate chains, when they are attached on proteins, they're called glycoproteins, and when they are attached on lipids, they're called glycolipids. And you know that the function of plasma membrane is the transport. So things get transported across the membrane. So plasma membrane is very important for the transport. So my question to, if we would have been in the class, I would have asked you, like, have you ever thought, like, if you have one small animal like a dog, you have another big animal like an elephant. So what do you think? Which animal will have, like, there's a dog, there's an elephant. Elephant is a big animal, dog is a small animal. So do you think the big animal like elephant will have big cells, or they will have more number of cells, right? So the question is, does elephant, which is a big animal, does it have, the size of the cell is big, or do they have more number of cells? So answer is they have more number of cells. So why they have more number of cells? Why don't they have big cells? The answer is there is a limitation to the size of the cell. And why there is a limitation to the size of the cell, which means that there is a limit to the size, which means after some specific size, the cell size cannot increase. And why? Because there is one ratio called surface area to volume ratio. Okay, surface area to volume ratio should be high. If this ratio goes down, then the transport will not take place across the membrane. Okay, so it's very important for the transport to take place across the membrane for this ratio to be high. Which ratio? Surface area to volume ratio should be high. Otherwise, transport will stop. And literally, what is the meaning of that? Surface area to volume ratio should be high means the surface area of the cell should be high. Only then transport can take place. If the cell becomes very big, its surface area increases, but its volume also increases. And volume, volume increases by the factor of 3. Area increases by the factor of 2. So if the size of the cell become very big, volume also increases. So the ratio is surface area to volume ratio. This is the ratio, surface area to volume ratio. If suppose that happens, volume increases, then this ratio decreases, and then transport cannot take place. So there is a limit to the size of the cells. Cells cannot increase beyond some specific size. So that is why the big animals, they don't have big cells, because if they have big cells, this ratio will decrease, because as the size increases, volume also increases. In the ratio surface area to volume is the denominator. It will decrease the ratio. And if the ratio decreases, transport cannot take place. So that is why the big animals have this kind of arrangement, which means instead of big cells, they have small cells and more number of cells. Okay, coming to the most important organelle inside the cell is nucleus. I told you yesterday also, nucleus is the brain of the cell. It is the main controlling center of the cell. Why? Because it has DNA. The genes are there, and genes are made of DNA, and you know what DNA is, right? So nucleus is covered by nuclear envelope, okay, and it is bilayered. And I told you yesterday also, the nature, it is a nuclear envelope is a membrane. So it has the same nature as the plasma membrane, and it is double. So this is your nucleus. Inside the nucleus, there is a region called nucleolus. And what is nucleolus? This is a region in the nucleus where rRNA is made. And what is rRNA? It is ribosomal RNA. Okay. And in the nucleus, inside the nucleus, there is chromatin. And we have already talked about what is chromatin. Chromatin is DNA wrapped very, very tightly around the histone proteins. So if you see a cell, when it is not dividing, you will see a dense mass, and that dense mass is called chromatin. But when the cells, they start dividing, and when they are in the mitotic phase, in the M phase, this chromatin basically gets condensed and takes the shape of the chromosomes. So this is your nucleus. This is covered by nuclear envelope, which is double layered. And these are the nuclear pores. Okay, these are the pores here through which things can move in and out of the nucleus. So you can see here, this is the nuclear envelope. It has an inner membrane, it has an outer membrane, it's bilayered. There are some pores here, and this is the chromatin. And the nuclear envelope is supported by nuclear lamina, which is a network of some proteins basically. So the shape of the nucleus is maintained by nuclear lamella, which is composed of protein. Okay, so we talked all about these things. Okay, this is the picture taken by the electron microscope. Okay, moving after the nucleus, let's talk about ribosomes. You know what are ribosomes? They're protein factories. So on the ribosomes, proteins are made, and we have already talked about ribosomes are made of rRNA and the proteins. And these ribosomes can be at two locations. They can be sitting freely in the cytoplasm. Those ribosomes are called free ribosomes. And There can be some ribosomes which can be embedded on the endoplasmic reticulum. I'm going to talk about endoplasmic reticulum too. And I told you in the last session what is endoplasmic reticulum. Reticulum means the network. Endoplasmic, it's inside the cell. It's a network of membranes inside the cell. And endoplasmic reticulum is again its extension of nuclear envelope. Okay, some of the ribosomes can be present on this endoplasmic reticulum, and those kind of ribosomes are called bound ribosomes. So there can be free ribosomes in the cytoplasm, there can be bound ribosomes. And you know what is the structure of a ribosome? It has a smaller subunit and it has a larger subunit. We have all talked about all these things. So this is the smaller, this is the larger. Okay, so there is a system of membranes in the cell, and that system is called endomembrane system. And what is included in that is basically system of the membranes. It includes your plasma membrane, it includes your nuclear envelope, endoplasmic reticulum, Golgi apparatus, lysosome, and some vacuoles. So these are all part of your endomembrane system. And I showed you yesterday in yesterday's lecture that how does different molecules move from like if they have to move from endoplasmic reticulum to Golgi, how do they move? Or if some of the proteins they have to be secreted outside, they all move with the help of the vesicles. So endoplasmic reticulum is basically the extension of your nuclear envelope, and it's basically a membrane. So it has the same structure as your plasma membrane. But I told you that what is the difference between the different membranes in the cell? It's only they have different proteins, and that determine the difference of the proteins in different membranes determines their different functions. Okay, the endoplasmic reticulum can be studied with the ribosomes. If it is studied with ribosomes, it's called rough ER, and if it lacks ribosome, it's called smooth ER. So you can see here, this is your nuclear envelope and how it's extending. Okay, this is endoplasmic reticulum. So endoplasmic reticulum is basically extension of your nuclear envelope, and you can see these sides, they are all embedded by ribosomes. So this is all rough ER, and this side you can see these sides they don't have ribosomes, so that is called smooth ER. And this is the part of endoplasmic reticulum which is responsible for making lipids, and this is the part which is embedded by ribosomes, that's responsible for making proteins. So this is your endomembrane system. You can see it has a nuclear envelope, it has an endoplasmic reticulum, Golgi apparatus, lysosomes, plasma membrane. Okay, so this is your smooth ER and this is your rough ER, and you can see how this is your membrane, this is the ER. Okay, and how this is a ribosome here, and ribosomes, on the ribosomes proteins are made, and how do they enter inside the lumen of the ER. And these are your ribosomes, and these are your ribosomes, and they are sitting freely in the cytoplasm. They're not on the ER. So these are all electron micrographs. Okay, these are pictures taken by electron microscope. So these are the functions of smooth ER. They synthesize lipids, they metabolize carbohydrates, they detoxify drugs and poisons, and stores calcium ions. These are the functions of smooth ER. And rough ER, you know what rough ER is, right? It has bound ribosomes which secrete glycoproteins, proteins covalently bound to carbohydrates, distribute transport vesicles, proteins surrounded by membranes, is a membrane factory for the cell. Golgi apparatus. I yesterday told you what is Golgi apparatus. Golgi apparatus acts like a post office. It basically, in the Golgi apparatus, some of the proteins needs to be folded more, they need to be processed more, and according to the signals they are brought, they are sorted, and they are sent to their destination. So first of all, what is the Golgi apparatus? It consists of flattened membranous sacs called cisternae. Those membranous sacs, they're called cisternae. They modifies products of the ER, the proteins which are made in ER, or even the lipids which are made in the smooth ER. It modifies them, manufactures certain macromolecules. Some macromolecules can also be manufactured here. Sorts and packages material into the transport vesicles. So this is these are the membranous sacs. These are the membranous sacs. Okay, and they are called cisternae. the side of the Golgi which is facing towards the ER, that is side called cis side. Basically that's the receiving side, and the side which is the shipping side, through which will be like after the processing, things will be the different molecules will be sent to the destination. That side is called the transphase, or the shipping side of the Golgi apparatus. So this is another picture of your Golgi. You can see how different molecules, they are coming from the ER, they get packaged in these vesicles, and they go get attached with the, so they bud off from here, and then they go get fused with the Golgi, and they will get processed in the Golgi, and then they will be again this part will bud off, and they will form a transport vesicles, and they will be sent to the destination. Now next organelle is a lysosome. What is a lysosome? Lysosome basically it's basically originates from lysosome is also part of endomembrane system. It basically, it's a kind of a membrane, it's a membranous sac, and it originates from Golgi. Okay, and it has some hydrolytic enzymes in it, and lysosomes are called garbage disposal system. Lysosome is a membranous sac of hydrolytic enzymes that can digest macromolecules. Lysosome enzymes can hydrolyze proteins, fats, polysaccharides, and nucleic acids, and lysosome enzymes work best at the acidic environment. So lysosomes is like the garbage disposal system, and they have some enzymes in them. They have hydrolytic enzymes in them. So anything which is not required in your cell, or something foreign enters into your cell, it is taken care by these lysosomes. And because these lysosomes have hydrolytic enzymes, they are going to like any organelle which the cell doesn't need, or any macromolecule or any bug enters into your cell, it will be just chopped by the enzymes of the lysosome. So here you can see this is some bug coming inside your cell. So it will be taken up by your cell by the process called phagocytosis, and it is going to form this kind of a vacuole, and it is going to fuse with the lysosome. Okay, this pink thing is or a purple thing is a lysosome. So this food vacuole is going to fuse with this lysosome, and the enzymes in the lysosome is going to digest. And similarly, like inside your cell, there are some organelles which get very old, and your cell doesn't need them. So the same thing happens. They get fused with the lysosome, and the enzymes in the lysosomes are going to chew the contents of the that organelle. It's going to basically degrade that organelle, and this is the way cell gets rid of any old or worn out organelles, and that process is called autophagy, which means when the cell eats its own stuff. Okay, so the lysosomes basically have to maintain acidic pH, because the enzymes in the lysosomes, they work at the acidic pH. Okay, so so these lysosomes, they have lot of these lysosomes, they have lot of these proton pumps. You know what proton pumps are? Proton pumps are the ones which transport the hydrogen ions. So through these the proton pumps, acidic pH is maintained inside these lysosomes. Proton pumps help maintain an acidic pH, while broken down macromolecules are transported out of the lysosome by the transporters. So transporters are some other membrane proteins. You know, we have already talked about what are transporters, and you know what are the proton pumps. Okay, the cells, like your plant cells, they have a big vacuole which is called the central vacuole. Okay, a plant cell or a fungal cell may have one or several vacuoles derived from endoplasmic reticulum and Golgi. But there are some other kinds of vacuoles too, like the contractile vacuoles, which are basically found in freshwater protists, and they help in the osmoregulation, which means they pump excess water out of the cells. Okay, and there are some food vacuoles which you just saw, which are formed by the process of phagocytosis. But the central vacuoles, which is a very big vacuole, so these vacuoles are also part of endomembrane system, These plant cells, they have a big central vacuole which is required to hold some organic compounds and water. And in some of the protists, which are some very small organisms, they also eukaryotes, they do have some contractile vacuoles by which they can do the osmoregulation. And food vacuoles you just saw. So this is the central vacuole in the plant cells. Okay, now the proteins which are made, let's come back to the protein and the ribosomes and how proteins are transported. So these proteins basically which are formed, they carry some signals within them. Like I said, the Golgi apparatus, it's going to sort them. The way the post office sorts the letters according to the zip codes, every protein also has some signals in them. Okay, if they don't have any signal, if they don't have any signal, it means that that protein is going to go in the cytosol. Means its position, its localized, its destiny is cytoplasm. If it is having a amino terminal signal, then it is going to go to the chloroplast or mitochondria. If it contains an internal signal, so these are the signals. Okay, if it has an internal signal, then it is going to the nucleus. And you know that the proteins, if they are made on the free ribosomes, then those proteins are functional in the cytoplasm. If the proteins are made on the ribosomes which are embedded on the endoplasmic reticulum, those proteins are either secretory or they become part of the endomembrane system itself. So you can see here, this we have already talked about. So these are the ribosomes. These are the ribosomes which are embedded on the endoplasmic reticulum. And once this protein is made, it enters into the lumen of the ER. Okay, it enters into the lumen of the ER, and then it is not fully made yet. It has to further go more processing. So with the help of vesicles, it goes to Golgi. There it gets more modified, and then it moves to the once it gets modified, then it again forms the vesicles. It buds off, forms the transport vesicles. Either it is going to be secreted out, or it is going to form the part of the membrane. Okay. Now let's see how the protein which is made on the ribosome which is embedded on the ER. Okay, the ribosome which is on ER, how does that protein enters into the lumen of the ER? Because if it does not enter into the lumen of the ER, then how will it go to the Golgi? Because that protein is not ready yet. Okay, so this is a ribosome which is bound to the ER. So let's see how it actually enters. You can see here the signal recognition particle. This is the signal recognition particle, binds to the signal sequence in the amino terminal end of the growing polypeptide chain. So this is your polypeptide chain which is being formed by this ribosome. Okay, so this is a ribosome, and this is a polypeptide chain, and it has a amino terminal signal sequence. So this signal recognition particle goes and binds here. Once that happens, translation process halts. After that, what will happen? So the SRP, which is the signal recognition particle, binds to the SRP receptor. So here is the SRP receptor. This is your SRP. Okay, it binds here on the ER membrane. So this is your ER membrane. It through this receptor, this SRP, through this receptor it binds here. The SRP receptor brings the ribosome to the transmembrane channel. Okay, this is the transmembrane channel. You know what channel is, right? The SRP receptor brings the ribosome. So this is a ribosome. Once it binds, SRP binds to the receptor, it brings the ribosome to the transmembrane channel, which is this. The SRP disassociates. So it's just getting, it's disassociating. The protein synthesis resumes. Okay, the protein synthesis resumes. It starts the process again, and the growing polypeptide chain is threaded through the channel. So once it leaves this receptor, once this SRP leaves this receptor, the translation process will start again, and this polypeptide chain will enter this channel, and it will enter the lumen. The signal peptide will be cleaved. The protein ends up in the lumen of ER, where it may remain, be transported to the lumen of another organelle, or be excreted out of the cell. Okay, this is the way these peptides, they enter the lumen of the ER. Now, as you know, some of the proteins, they get embedded in the transmembrane. They get embedded in the membrane, and those proteins are called the transmembrane proteins. So, how does that take place? Let's see that. So, this is your ribosome, and it is this is the ER. Let's see how does that take place. Proteins with signal anchor sequences. So, those proteins which get embedded in the membrane, they have signal anchor sequences. Okay, proteins with signal anchor sequences are threaded through a channel in the ER membrane until the signal anchor sequence is encountered. So the proteins with signal anchor sequence, so this is a protein with the signal anchor sequence. This is a signal anchor sequence. Okay, so anchor sequence, anchor is something which attaches. Okay, it has a signal to anchor into the membrane. Proteins with signal anchor sequences are threaded through a channel in the ER. This is the channel in the ER until it encounters this signal anchor sequence. The ER channel releases the protein into the membrane. So once it encounters this anchor sequence, it is going to release this protein into the membrane here. Okay, when translation is complete, the protein remains in the membrane. Okay, so it will release the protein into the membrane, and it will the translation will continue, and once the translation is complete, this protein remains in the membrane. So this is the way the proteins get anchored in the membrane. Okay, the next organelles are mitochondria and chloroplasts. Mitochondria and chloroplasts, they are the two organelles. Mitochondria is responsible for the extraction of the energy from the food we eat, and chloroplasts, as you know, they are in the plant cells. These are the organelles responsible for photosynthesis. And one more organelle we need to study are called peroxisomes. They are oxidative organelles. So mitochondria and chloroplasts, they have similarities with bacteria. Okay, and what are those? Both are enveloped by double membrane, contain free ribosomes and circular DNA, grow and reproduce somewhat independently in cells. So there is one theory called endosymbiotic theory. According to this theory, these chloroplasts and mitochondria, they are now organelles in the eukaryotic cells. According to this theory, mitochondria and the chloroplasts, they are basically prokaryotic organisms. Okay, so you know what evolution is. So it's considered that mitochondria is an aerobic bacteria and chloroplast is a photosynthetic bacteria. So in the evolutionary track, billions of years back, this photosynthetic bacteria, which is chloroplast, and the aerobic bacteria, they started living in the eukaryotic cell. And while they were living in the eukaryotic cell got the ability to do the oxidative phosphorylation or the respiration. I mean, it got the ability to extract the energy from the food. And the photosynthetic bacteria, when it started living in the eukaryotic cell got the ability of photosynthesis. And on the other hand, both these bacteria, photosynthetic and aerobic, they got a nice home to live, which is a eukaryotic cell. So this is a situation called symbiosis. It's an association in which both the partners, they benefit. So this is the theory. This theory says that the mitochondria and chloroplasts, they are basically bacteria. They're prokaryotic organisms. And here is a list, because there are some evidences on the basis of which this theory was formed, and those the similarities are here. The chloroplast and mitochondria, they both have the double membrane like the bacteria, and these organelles, they have their own DNA and they have their own ribosomes. Okay, which is something really special. Okay, according to this theory, you can see here, this is a eukaryotic cell, and this first of all, the bacteria, this is a eukaryotic cell. It got the aerobic bacteria, it started living, and it became the mitochondria. And then this photosynthetic bacteria started living in the eukaryotic cell and became a chloroplast. Okay, now let's study the structure of mitochondria and the chloroplast. So this is mitochondria. Mitochondria has two membranes. Okay, one is the outer membrane and then there is the inner membrane. So the membrane is bilayered, and between the outer and inner membrane there is an intermembrane space, and the inner membrane is invaginated like this. Okay, this is invaginations of the membrane, inner membrane. This inner membrane is invaginated so as to increase the surface area, and these invaginations are called cristae. And why they are invaginated to increase the surface area? Because here are some very important enzymes here which we will study in the chapter on cellular respiration, that they are very important in the cellular respiration. And this part of the mitochondria is called matrix, and in the matrix is DNA and ribosomes. So this is the basic structure of mitochondria. And next is your chloroplasts. So chloroplasts, they also have two membranes, outer and the inner ones. And inside the chloroplast, so this is the outer membrane, inner membrane, they have these kind of membrane, they have kind of these, they call the thylakoids. Thylakoids are membranous sacs, and they are stacked to form a granum. So these are thylakoids. Okay, these are thylakoids. These are like membranous sacs, and they are stacked on each other, like you must have seen the stack of pancakes. So these thylakoids, which are membranous sacs, they're stacked on each other and they form the granum. Okay, and here is, in this area is all jelly kind of a material here. It's called stroma, and in the stroma are the DNA. This is the DNA of the chloroplast, and these are the ribosomes. So this is the basic structure of the chloroplast. And the last organelle is peroxisomes. Peroxisomes are organelles which are bounded by single membrane, and they do have some enzymes in them. Okay, like lysosomes, they are membrane bound vesicles that enclose enzymes. Now the peroxisomes contain enzymes whose action result in hydrogen peroxide, and you know what hydrogen peroxide is? A toxic molecule, and hydrogen peroxide is broken down to water and oxygen by another peroxidase enzyme called catalase. So peroxisomes are prevalent in cells that are synthesizing and breaking down fats. So peroxisomes are basically the enzyme. These are the membrane enclosed vesicles that enclose the enzymes, and they help in the breaking down of the fats. Okay, so this finishes this lecture. I hope it's clear to you. If you guys have any question, you can always send me email, and I will see you on Monday, and I will be assigning you the Launchpad assignment from chapter number 5, so you guys can do that. Okay, bye.